Hi, this is Dr. Muneer Ahamed from South Australian Health and Medical Research Institute. I'll be talking about preclinical evaluation of our PET ligand targeting the endocannabinoid alpha beta hydrolase domain 6. So the endocannabinoid system consists of two major cannabinoid receptors, CB1 and CB2, and also lipids such as 2AG and AEA, which also acts as endogenous ligands for the cannabinoid receptors. Plus, there are also enzymes which either synthesize or degrade these lipids. For example, 2AG signaling function is terminated by MAGL and ABHD6. So the ABHD6 inhibition leads to a marginal increase in 2AG levels in the brain, which has proved to be beneficial in many CNS disorders and also in some of the non-CNS disorders as well. The regulation of 2AG levels by ABHD6 is emerging as an attractive strategy with multiple therapeutic benefits. So as I said before, the 2AG signaling function is hydrolyzed by ABHD6. So what happens here is there is an increase in 2AG level when you inhibit ABHD6. This also means that there is a retrograde activation of the cannabinoid receptor. So this is also avoids any 2AG overload in the brain. In other words, it doesn't desensitize CB1 receptor when you inhibit ABHD6. That's not the case with MAGL. So this is also beyond a classic direct activation of cannabinoid receptor, and hence is an attractive strategy to target endocannabinoids. So on the other hand, you can see the inhibition also leads to a decrease in the prostaglandins, such as pro-inflammatory and pro-oncogenic prostaglandins. So ABHD6 has multiple physiological and pathological roles. For example, in the brain, it acts as an endocannabinoid signaling pathway and also has direct relevance to neuroinflammation. It is also one of the major 2AG hydrolase in microglia and macrophages. And for non-CNS point of view, it is also a major enzyme in pancreatic beta cells and also has direct relevance to glucose-stimulated insulin secretion. But we are particularly interested in looking at neuroinflammation. As you can see, the blockade of 2AG is found to have an increased 2AG CB2 signaling axis, and also it inhibits immune cell infiltration into the CNS, and it exerts an anti-inflammatory effect. Particularly, we are also interested in studying epilepsy by targeting ABHD6. Inhibition of ABHD6 has shown anti-epileptic activity, and also it significantly reduced seizure frequencies in both chemically and genetically induced animal models. So our target molecule was rationally designed. It contains a carbamate group and also a thiadiazine group. As you can see, the aryl group nicely fits into the axianion hole, and also the piperidine or the morpholin moiety fits into the L-shaped side of the enzyme. As any serine hydrolase enzyme, so this ABHD6 also have a catalytic triad, and studies indicate that mutagenesis of serine amino acid leads to a lot of loss of catalytic activity. So besides application of these new tracers in imaging ABHD6, so it also has application in drug discovery, such as validating a new biological target, or we can validate a new tracer. We can also find out drug dosing and also receptor occupancy in multiple species. We can also study target selectivity and also study species differences. As you can see, there are a number of small molecule drug approvals in 2020 FDA drug approvals, and most of this drug discovery effort, PET imaging forms a major contributor. So first we did a comprehensive pharmacological study to see our targeting ability and selectivity for our compounds. So we did the activity-based protein profiling in whole brain membrane proteome for our two compounds with three different concentrations. As you can see on the panel, at about 100 nanomolar, our target compound 11 only inhibit ABHD6 and does not do any inhibition for MHGL or ABHD12 or FA or any other serine hydrolases. And also, you can see that so JZP-M11 did not act as an agonist or inverse agonist at any of the CB1 or CB2 receptor. So this whole points towards a complete selectivity towards ABHD6. So next we did radiosynthesis using copper-mediated aromatic fluorination. So it's a well-known method for radiofluorination of aryl groups. The only difference we did not use any water or cryptofix carbonate for fluoride elution. So instead we used dry methanol and tetraethyl ammonium carbamide. So we got about 4% radiochemical yield with sufficient molar activity for any neuroimaging. So once we had the tracer, we did the in vivo imaging of ABHD6 to study binding kinetics and also specific binding in different tissues. So as you can see in the brain, so there is a rapid uptake of the tracer and also slow washout, indicating an irreversible binding. We also see specific uptakes in heart, kidneys, and liver, as indicated by blocking studies. And similarly, we also see a block in the brain. So we also did a blocking with a structurally non-related compound, which is WWL70. This sort of experiment is pretty standard to validate specificity of a particular tracer. So in this case, we see specific binding in heart and kidneys, those peripheral tissues, but we did not see any blocking in the whole brain. And this does not surprise us because it's not very uncommon for the CNS PET traces, as once you have a peripheral binding effect, so you block all the peripheral binding sites, and your radioligand concentration in plasma will be quite high, and you will get a lot of the PET tracer to get in the brain. And so I've shown one literature example where Victor Pike has showed in his imaging studies that so when you do a baseline, so you don't see any brain uptake, but when you do a blocking study at the early time point, you can see there is an increased brain uptake. So this shows that there is a peripheral binding effect. We also did ex vivo autoradiography study to understand regional brain distribution. You can see, so we got pretty nice images of frontal cortex, hippocampus, and hypothalamus. And also we can show that uptake is specific for these regions. And we are also surprised to see there is a direct correlation between the mRNA expression level on the Allen's Brain Atlas for ABHD6 and our autoradiographic images, particularly for frontal cortex and hippocampus and hypothalamus regions. Next, we did in vitro autoradiography to study dose-dependent blocking. So in this case, so we use three different concentration of the structurally non-related inhibitor WWL70 at 100 micromolar, 50 micromolar, and 10 micromolar. And you can see we obtain 55% and 30% and 13% blocking effect in those concentration, which indicates specific binding of this particular tracer. So in conclusion, so we have developed the first PET ligand to image ABHD6 in vivo. This is also a highly potent and selective ABHD6 inhibitor. So we showed brain proteome-wide selectivity for ABHD6, and we also showed that this is not a substrate for either of the cannabinoid receptor. So we saw high brain uptake with slow washout in the mice, showing irreversible binding. ABHD6 specificity was established by both in vivo and ex vivo blocking experiments. And currently we are evaluating a number of experiments in non-human primates in Belgium with Professor Guy Bowmans. And we are also exploring potential application in neuroinflammatory and neurodegenerative disorders with this particular tracer. I would like to thank a number of people here, Chris, Idris, and Rajiv during my stay at CI, where most of this work was done. And for endocannabinoid pharmacology and medchem help from Finland and Auckland with Yuva, Jayendra, and Natasha. And Karine for all the animal imaging work, and Damien for radiochemistry help. And thanks for listening.